And at the end of the year, so we're going twice this semester only, then we will go in the spring 3 times, and at the end of the year, we bring them to campus. So none of these are pictures of them on campus. But they come spend the day at Roberts. They go to the gym and do activities with PE students. They eat in the cafeteria, and they do a biology activity, a chemistry activity, and they leave thinking that college is the greatest thing that's ever happened to them because they can eat as much pizza as they want. Okay, so that's, I mean, literally their takeaway is the cafeteria is the greatest thing that's ever happened. But for some of them, it's that, you know, it's their first time exposure to the college campus, what college life might be like, and seeing what college would be like. We get out for biology, we get out the human lungs and let them like inflate the lungs and dissect a cow eye and stuff. It's a lot of fun. It is a commitment in that when I ask people, and if you come to a meeting today at 11 where we talk about it, you're getting a little bit of information here. It's a commitment where you need to commit to do it, because if you commit to do it and you go and you're not there, they're gonna wonder where you are and they're gonna ask about you by name, because they're gonna remember you. So it's really important that if you make this commitment that you come, because it's a relationship that is being built with these students, and it's important that we maintain that relationship and that trust with them. Okay, so that's today at 11 a.m. in the Science Lounge. If you come at 11 a.m., you're not committing. We're gonna go over the lesson that we're doing on October 18th, but you're not necessarily committed. Okay, so you can come hear more about it and see what a lesson might look like. The Beyond Conference is Monday, which means we don't have class Monday. I keep getting questions about it that I don't have answers for, but you're supposed to go to stuff. There's some link where you're supposed to register by 1 o'clock today, is what they said. So you got an email this week that said it was clarifying it. That clarifying email said you need to register by 1 o'clock today. So there's like an opening session and then there's a workshop. That's what you need to register. There's choices for the workshops. Those are the things that you need to register for, the 2 workshops. There's one before lunch and one after lunch. Those are the things you have to register for, the like opening and the closing things you don't have to like register for. And you're using the Chapel app to register your attendance on it. That's all I know. Sorry. The clarifying email didn't clarify things for me, but that's what I know. So, chapter 9 homework, because we don't have class on Wednesday, it's gonna be on Monday, it's gonna be due on Wednesday. I just posted it last night, so if you're thinking, hey, it's not there, it is there. We're doing another outline because the feedback I got on the test was that a lot of people were using them anyway. I think they're helpful. We're not gonna do them every time necessarily. I posted it last night, and you should have gotten your chapter 7 one back with a little bit of feedback on it, so that you can be kind of what we're looking for. And also speaking of things that are coming up, advising is coming up. You should have gotten an email from your advisor talking to you about getting ready for spring 2025. Some of you are my advisees. You should have gotten an email from me. In that email, it's going to tell you how to schedule a time to meet with your advisor. In order to register for spring classes, you have to meet with your advisor. So you have to do something about this. Okay. Seniors start registering October 21st. I think freshmen start registering
the 30th.
Thank you. I'm like later, October 30th. So in order to register, you have to meet with your advisor because they have to approve you to register for the courses that we're selecting. So you can't put it off forever. Look for that email from your advisor. If you didn't get an email from your advisor, you can feel free to email them because they were supposed to have done this already. Okay? That's all my procedural stuff. There's only 6 minutes of announcement stuff. Okay, so we're gonna dive into chapter 9. Just some things to think about as we are starting a new chapter and a new unit. The next test is gonna be on chapter 9 and 10. I have graded not all the tests, but some of the tests, and you need to be active in learning, is what I'm saying still. Okay, when you come to class, you can't expect to passively take in the information. You need to be doing something, like writing it down. Write it down, print the PowerPoints, you can write on the diagrams. There are lots of diagrams in this chapter. And when I say lots of diagrams, I mean lots of diagrams. And having them on paper in front of you so you can write on them is helpful. I promise. I'm not just saying this, and I would keep saying it, and eventually it might sink in. Okay, so chapter 9 is about cellular respiration and fermentation. Before we go into that, I don't like to go into the details of cellular respiration and photosynthesis without talking about the big picture. This chapter 9 is super detailed. Okay, up until this point, you might think we've been detailed. We've not been super detailed. We're going super detailed now. But I want you to keep, as we're talking about the details of cellular respiration, I want you to be able to keep the big picture in mind. Because the big picture is helpful in understanding why we care about the details of it. So here we see the cycling of energy and carbon. Sometimes we call this the carbon cycle or part of the carbon cycle through the environment. So this is our big picture. So we have two main reactions that are taking place in this big picture. We have photosynthesis and we have cellular respiration. So we are going to, this is chapter 10. Everything above the line is chapter 10. Everything below the line is chapter 9. So chapter 9 is focusing on cellular respiration, a little bit of fermentation, and chapter 10 is going to focus on photosynthesis. Again, this is your whole next test is just on these two chapters because there's so much detail. But this is the big picture. Okay, so what do you notice in this big picture? We'll start with cellular respiration. We're taking organic molecules like the example we will use as we talk about it is C6H12O6, which is what? Glucose. What kind of molecule is it? It's carbohydrates. Okay, so it's a carbohydrate and we're going to combine it with oxygen. Because what are we trying to make? Why do we do this reaction? What is the point of cellular respiration? We are trying to make ATP. Okay, this is the point. It's the stars. It's already in a star shape. This is why we want this reaction to happen. Because we need ATP. Okay, so if we're taking glucose and oxygen, I want to think really big picture. How do you get glucose? Where does glucose come from for you? Food, we eat. Okay, so the source of this, it is not always glucose when we say organic molecules. Other organic molecules can also be in this reaction, but this is coming from food. So what system of your body is getting this to you? The digestive system, when you eat, breaks down your food into simpler molecules. How does it get to your cells? So you're eating food, it's going to your stomach, it's getting digested in your intestines. How does it get to your body? It's absorbed into your intestines, I agree. Into what system? Maybe that's a better question. Yeah, it's going to be transported by the circulatory system to all the cells because every cell in your body needs to be able to carry out cellular respiration. You eat the food, but it's got to get everywhere. So the digestive system breaks it down and the circulatory system takes it to the cells. How does it get into a cell? What process moves glucose into a cell? Chapter 7. What kind of diffusion? Is it simple diffusion? It's not simple diffusion because simple diffusion doesn't work very well for polar molecules. What kind of diffusion is going to get it in? There's not a lot of other choices. Facilitated diffusion. So it's going to enter the cells. So it's going to get to your cells. By facilitated diffusion. This is why you eat. You eat because you need to carry out cellular respiration. That's why you eat. Okay, most people understand that you eat because you need energy. Okay, on the other hand, we also need oxygen. How does oxygen get into your body? You breathe it. And when you breathe it, what system takes it in? So oxygen's coming in through the respiratory system. How's it getting to the cells? You breathe, it's going into your lungs. How's it getting to your toes? Circulatory system. So the circulatory system is going to carry it to your cells. How's it going to get into a cell? Somebody said it. Diffusion. Oxygen is a non-polar molecule, so it can do simple diffusion. So it's getting into the cells through simple diffusion. So we always portray this reaction as glucose plus oxygen makes ATP. You'll see in this chapter that is true, but it's a much more long and convoluted process. It's a long multi-step metabolic pathway in which every step is catalyzed by what? Enzymes. Okay, are you going to learn all those enzymes in this class? No, be thankful. You are not learning them all in this class. Wait for biochemistry. Biochemistry, you can learn all the enzymes that are involved. Probably not even all of them still. Okay, so we're taking this, we're producing ATP. ATP produces power cellular work. Where is this taking place? In the mitochondria for the most part. Okay, so this takes place in the mitochondria. And again, we'll relate the structure of the mitochondria to the function of the mitochondria. Does this happen in prokaryotic cells? Not fully, but they still need ATP and energy. So they don't have mitochondria because they don't have membrane-bound organelles. They are going to carry this out just with cellular membranes alone. So without the membrane-bound organelles of the mitochondria. This reaction is this kind of reaction. What kind of reaction is this reaction? It's an exergonic reaction, so our reactants have more energy than our products because we're releasing that energy as ATP. So energy is released. Again, because we're changing forms of energy, sometimes we lose energy. And as we do this cellular work using ATP, we release heat energy. So heat energy is still energy, but not as useful in terms of energy. What kinds of organisms do cellular respiration? You guys are making me nervous. What kinds of organisms do cellular respiration? All of them to some degree. Okay, plants do cellular respiration. They have mitochondria. They have energy needs. We tend to not think of plants as needing energy because they just kind of stand there, grow there. They aren't mobile, totally mobile. They do have some ability to move some ways, maybe not their whole body, but plants do this, animals do this, protists do this. Fungi, all of those, and prokaryotes. I don't want to say that they all do. Fungi and protists, fungi and prokaryotes don't necessarily do straight up cellular respiration, but they do some kind of reaction that produces ATP that's got some relationship to cellular respiration. Okay, so when we do cellular respiration, the reactants are glucose and oxygen or some other organic molecule. What are we making from that? What are the products of it besides ATP? ATP is the goal, but there are also some other products. What else is made? Carbon dioxide and water. So these are the waste products of cellular respiration. This is the products. Reactants are glucose and oxygen. The products are carbon dioxide and water, which we call waste products and or byproducts. Okay, water is a byproduct in that it's not harmful to us, so it's not causing a problem. Carbon dioxide is not something that we want to build up, so that's a waste product. How do we get rid of carbon dioxide? We breathe it out. So this is like the reverse of how we get oxygen. It's going to diffuse out of the cell through diffusion. It's going to go into the circulatory system. It's going to be carried back to the respiratory system, and we're going to breathe it out again. Okay, you're literally breathing out the carbon molecules that you ate. Not immediately, but you're breathing out the carbon molecules. Water, we don't really have to worry about getting rid of it because it's helpful for our bodies. So that's the products of cellular, this is the reactants of cellular respiration. These are the products of cellular respiration. But you'll notice, what are they for photosynthesis? These are the products of cellular respiration, but they are the, somebody said it. They're the reactants. We're going to switch colors. These are the reactants for photosynthesis. So the products of cellular respiration and the reactants of photosynthesis are the same thing. What kinds of organisms do photosynthesis? Plants and some protists and some prokaryotes, but we're not going to worry about that. So this is a reaction that's primarily taking place in plants or other autotrophs. So how does a plant get carbon dioxide in? You haven't talked about plants in a long time in your life. They do. They breathe it in. They have little holes on their leaves or sometimes on their stems, depending on the kind of plant, called stomata, that are like little mouths that let oxygen in. And I mean, sorry, well, oxygen too, but let carbon dioxide in. What process is going to let it into a plant cell? Diffusion, just like in an animal cell or a plant cell for respiration. So this is coming, oh, hello. This is coming in through, it doesn't want me to write anymore. Why are we not friends anymore? So this is coming in through stomata and diffusion into the cell. How do plants get water? From the roots. So water is coming in through the roots. What processes allow water to move up a plant? Some osmosis. That's more getting into the cells. Way back in chapter 3. Cohesion and adhesion create the forces that will allow the plant to move up. So this is, and osmosis, help it to move throughout the plant. Okay, this is an exergonic reaction. It's also what we call catabolic, which I didn't say before, sorry. This reaction, the reaction of photosynthesis, is the opposite. What's the opposite of exergonic? It's endergonic. What does that mean in terms of energy? The reactants have lower energy than the products. So reactants are lower than the products. So what has to happen for this reaction to occur? We need to get what? We need to get energy. Where do we get it from? The sun. So there has to be an input of energy. So the energy input comes from the sun. If it's endergonic, it's not catabolic, it's what? Anabolic. It's building up reactions. We're making something more complex. We're taking these simple reactants and we're making more complex products. So these reactants of cellular respiration are the products of photosynthesis, which I can't remember. So we call this the carbon cycle, or this is part of the carbon cycle, because we're moving carbon around. We're taking carbon from carbon dioxide, we're making carbohydrates, we're breaking down the carbohydrates, we're making carbon dioxide. But the point of this reaction in terms of us, the reason we care about this reaction so much is because it's producing ATP for us. This is the big picture. Okay? We're going detailed to talk about cellular respiration and fermentation. Okay, so chapter 9 is going to focus on the bottom half of that diagram. Chapter 10 is going to focus on the top half of that diagram. We're talking about cellular respiration and fermentation, and I'll be honest with you, I don't know why there are pictures of puffins. There's no puffins in your book, and I don't know why, but I just skeptical. Those companies are cute and they do cellular respiration. Maybe it's because they're extending energy by flying. It appears to be eating something, so it's getting organic molecules. I don't know. I think the old edition of the book probably has puffins. Your book had, I forget what the picture was. I did look at it. Okay. So before we get into the details of cellular respiration, we have to do some overview of kinds of reactions. So we keep coming back to chemistry. Okay, you're never going to get away from chemistry, biology, sorry. I know that doesn't always bring joy to people's hearts. I don't know if in chemistry class you have touched redox reactions yet. My guess is no. You did? Oh, then we can just skip this whole slide, right? Oh wait, everybody's not in chemistry. Not everybody's in chemistry, I get that. Okay, so when we talk about redox reactions, redox is short for what? Reduction and oxidation. So these are reduction and oxidation reactions that go hand in hand. And what we mean by that is it's a way to transfer electrons. So by transferring electrons and moving electrons around, we can release the energy from the chemical bonds. Remember, we say that chemical bonds are potential energy. So as we move these electrons around, we transfer them from one molecule to another. That movement of electrons releases some of that energy. And I'm assuming at some point, if you've talked about redox reactions, you've heard there's two ways to remember it. Okay, and I will tell you that still at my age, which is way older than you, I have to think about this every time when I think about reduction and oxidation, because I'm not a chemist. So you can think of this, you can remember this as oil rig. This is how I remember it. This is how we remembered it back in the 1900s when oil was king. Okay, oil rig, what does that mean? Okay, nobody remembers it that way anymore. Yeah, so when we're talking about transferring electrons, you can either lose electrons or you can gain electrons, and one of them is one and one of the other. So you have to remember that oxidation is loss. So an oxidation reaction loses electrons. When an oxidation reaction occurs, something is losing an electron, and reduction is gain. So when you reduce something, it gains electrons. It's counterintuitive. That's why every time I look at a reaction, I have to say oil rig. Okay, got it. Oxidation means losing electrons. Reduction means gaining electrons. Did you all learn the other one then? Leo the lion says grr. Do you not know either of these? Oh, whoops, sorry. Okay, Leo the lion says grr. Is kind of the opposite, where loss of electrons is oxidation and gain of electrons is reduction. So I didn't draw a line and stuff. I'm not that artistic, sorry. Some people draw lines. So loss of electrons is oxidation, gain of electrons is reduction. Whichever one makes you happy, whichever one gets it into your brain, whichever one you repeat to yourself when you're taking a test and thinking about electrons is fine. I'll be saying oil rig, you can be growling like a lion. Don't do it too loud during a test, but either way is fine. Okay, so this is showing you a generic reaction. I want to give you a real non-biological reaction. So if we have sodium and chlorine, we know what kind of bond are they going to form? An ionic bond, because they are, I almost said organisms. Wow, we're not organisms. They are atoms that have electrons, 1 electron in their outer shell or 7 electrons in their outer shell. So they want to either gain 1 or lose 1. That makes them happy. So sodium has a tendency to do what? What kind of ion does sodium become? It tends to lose to become a cation, which is positive or negative. because we love cats, it's positive. Even though they're destructive and catabolic, it's confusing. Okay, so in this is like a step to making a salt. Sodium is going to lose an electron. So it is what? Oxidized or reduced? It's going to be oxidized. So it's going to lose an electron, which gives it this positive charge. When it loses an electron, who's it giving it to? It's giving it to chlorine. So chlorine is what? It's reduced because it's going to gain an electron. So what kind of charge is it going to get? It's going to be a negative charge. It's going to be an anion. Okay, so in this reaction, then they will come together and make salts in an ionic bond. But this reaction, we have sodium being oxidized and chlorine being reduced at the same time because redox reactions go together. Reduction and oxidation happen at the same time. So that's a non-biological example. This is just showing us a more generic form of an equation. So we have this one, which already has a negative charge. So it's molecule X, which has an electron attached to it. And it's going to combine with Y to form. So these are the reactants. The products are X and Y with an electron stuck to it. So which one is getting oxidized? Why is X getting oxidized? It had an electron, now it doesn't have an electron, so it lost an electron. So X is oxidized. Y, on the other hand, doesn't have an electron. Now it does have an electron, so it is being reduced. Because the electrons are being transferred. Okay, this is a non-biological example. This is like a math equation kind of example. And this is the generic equation for cellular respiration. It doesn't look as pretty in that there's no like negative and positive charges floating around. So it's a little harder to tell which one's being oxidized and which one's being reduced because we're moving things. In addition to the electrons, we're also moving hydrogen ions. So we've got some hydrogen stuff going on too. So in this case, we've got glucose. And glucose is being converted. It is being oxidized. It's losing electrons. And it's going to form carbon dioxide in terms of electrons. So the electrons, it's losing electrons. As it loses electrons, it becomes carbon dioxide. It's also losing hydrogen ions. So it's losing and we'll talk about why hydrogen ions often move with electrons. Oxygen, on the other hand, if glucose is being reduced, sorry, I just said it. If glucose is being oxidized, then oxygen must be being, reduced. What a surprise. So it's going to gain electrons. So this oxidation of glucose is going to transfer the electrons to a lower energy state that provides energy. This oxidation provides energy for an enzyme known as ATP synthase. We know it's an enzyme because it ends in, and what's it going to do based on its name? What do you think it's doing? It's going to synthesize ATP. ATP synthase makes ATP. So this oxidation provides the energy that is needed to make ATP synthase happens. Okay, we're changing, in this case, this oxidation is changing the carbon-hydrogen bonds that are found in this carbohydrate. We're changing them to carbon-oxygen bonds. Carbon-hydrogen bonds are nonpolar, but carbon-oxygen bonds are polar because oxygen is more electronegative than carbon. So this reaction, this whole reaction happens in a stepwise fashion, and enzymes are catalyzing it to reduce that activation energy to make this reaction possible. So glucose becomes oxidized. This makes it confusing, but we call it a reducing agent. Because it's giving away electrons, it's helping something else become reduced. So even though it's becoming oxidized, it's helping to reduce something else. So it's a reducing agent. Oxygen, on the other hand, is the oxidizing agent in this reaction, which kind of makes sense because oxygen and oxidizing sound the same. I think I said everything. So this whole thing overall, again, big picture is a redox reaction. And we're going to trace those electrons, but we're also going to keep track of everything else that is happening too. Before we really get into the reaction itself, we also need to talk about the concept of an electron carrier. So we're talking about transferring electrons. It's generally not just electrons floating around here and there doing whatever they feel like. Okay, the electrons are being moved. They're part of a molecule that is carrying them that is an electron carrier. So again, a lot of times when we talk about electron carriers, it's not just they're carrying electrons, they're also carrying hydrogen ions. So this is an example of an electron carrier. This is NAD+. So if it's positive, is it going to lose electrons or is it going to gain electrons? If it's got a positive charge, it already has lost electrons, right? It's already lost, so it's probably wanting to gain them. This is the oxidized form because it has already lost electrons. So it's NAD+, which stands for nicotinamide adenine dinucleotide. You don't need to know that. But you should look at the structure of this and say, hey, it looks like something I've seen before. What does it look like? Something that you've seen before. We've got a house. We've got a driveway and we've got a pool. What is it? We've got a sugar attached to a nitrogenous base and a phosphate group. It's a, I'm hearing muttering, but not loud enough that anyway. ATP is an example of one of these two, yes. Not a phospholipid. It's got a sugar and a nitrogenous base and a phosphate group. So it does have a phosphate group, but what? A nucleotide, yes, which makes up a nucleic acid. So this is what we call a dinucleotide because it's got two pools, two sugars, and two nitrogenous bases. That's why it's nicotinamide adenine dinucleotide. It's two nucleotides stuck together. So it's a familiar format in terms of that. So this reaction, this molecule can carry electrons. Okay, it's oxidized, so it's down an electron. It has the ability to carry electrons. And it gets those electrons from food, but it doesn't just get straight up electrons. It also gets hydrogen. So it gives you an apple. It's good apple season. I should ask you what your favorite kind of apple is. I'll ask on Wednesday. You should have a favorite kind of apple. That's not a Red Delicious, so as long as you don't put Red Delicious, you're okay. So when you eat, you take in things like hydrogens and electrons. And this food is going to donate 2 electrons and 2 hydrogen ions. 2 of those electrons and 1 of those hydrogens is used to be able to reduce NAD plus to NADH. So NADH is the reduced form, NAD plus is the oxidized form. I like to think of them as full and empty. So which one is going to be full? Which one is a full electron carrier? Which one got the electrons? NADH just took these electrons. So this is the full electron carrier. Over here, NAD plus has the ability to take electrons, but it doesn't have extra ones. It doesn't have a full car seat. I don't know what they're carrying them in. It doesn't have a full complement of electrons. So this is an empty electron carrier. So there's an enzyme, of course, that makes this happen because we like enzymes. Dehydrogenase is the enzyme that will take the hydrogens off the food molecule to transfer to the nicotinamide. And then that also leaves 1 extra hydrogen because remember we had 2 hydrogens here because these guys are traveling together. So we're only giving 1 hydrogen to the NAD plus to make it NADH. So we've got an extra hydrogen hanging out over here. This hydrogen is going to combine with oxygen and electrons to make water, which is one of the byproducts. So we have the electron carrier NAD plus empty, full is NADH because it's been given electrons. Another one is FAD and FADH2. So this is the oxidized form of FAD. So if it's oxidized, it's lost electrons. So this is an empty carrier. And the reduced form has gained electrons. So it is a full electron carrier. You don't need to like memorize the chemical structures of these or anything. You just need to understand that when you see NAD plus, it's empty. When you see NADH, it's full. Because as we go through the steps of cellular respiration, you're going to see that happening. We're going to take full electron carriers and we're going to empty them, or we're going to take empty electron carriers and we're going to fill them. Same thing for FAD. FAD is empty, FADH2 is full. And we're going to go back and forth between these different forms, moving these electrons around because that's going to provide energy and release energy that's found in the organic molecules. So if you think of the movement of electrons through this reaction of cellular respiration, the electrons are going to go from glucose. They're going to get moved to NADH. From NADH, they're going to get moved down the electron transport chain, which we'll talk about in more detail than you might want to. And then eventually the end product is they're going to be accepted by oxygen to make water. So we're going to think about how carbon moves through this whole thing and oxygen, but we're also thinking about how electrons move through this whole thing. So electrons are going to be taken from glucose. They're going to be used to fill an NAD plus to make it NADH. And then the NADH is going to take it to the electron transport chain, which is the whole chain that transports electrons. And then eventually it's going to be accepted at the end by oxygen to make water. So electrons have a flow through this whole process too. Okay, so we are going to start. And we're probably just going to get through the overview of cellular respiration because, you know, I've only been talking for 40 minutes. Sorry. So this is an overview of the stages of cellular respiration. This diagram is in your book, and they're very careful to maintain color coding. That makes me happy. I love color coding. So The stages of this, as they talk about it in the book, it's always pictured with the first stage in blue. Then this is like a lighter red and a darker red and a purple. All of that is kept so that you could follow that organization. So it's color coded in all the diagrams and in your book. So we start at the beginning, the first stage of cellular respiration is glycolysis. So everything in the book that talks about glycolysis, any diagram or anything is going to show it to you in blue. What does the word glycolysis tell you about it? It's the initial breakdown of glucose. So glyco, carbohydrate or glucose, and lysis to break down. So the name tells you what it is. It's the breaking down of glucose. We're going to turn glucose in glycolysis. We're going to turn glucose. How many carbons are in glucose? 6. So remember I said we're going to keep track of electrons. We're also going to keep track of carbons. So we've got a 6 carbon molecule. We're going to break it down into pyruvate, which is just another carbon molecule. It has 3 carbons. So we get how many? We had 6. So how many pyruvates are we going to get? If pyruvate has 3 carbons, we're going to get 2 pyruvate molecules. Looking at this diagram, where is this happening? What part of the cell is this happening in? Not yet. Okay, this is definitely mitochondria. Okay, over here, we have a mitochondria. This is the mitochondria, but we're not in there yet. So where do you think we are? Yeah, we're just in the cytoplasm, the cytosol. So from this reaction, we're going to get a little bit of ATP, which is the point of the reaction. We're also going to move some electrons. So the electrons are going to move. We're going to make some NAD plus into NADH. So we're going to fill some electron carriers. These electrons then are going to go from here all the way down. They're going to skip to this step. And we're going to get this ATP formed. This is through what we call substrate level phosphorylation. So phosphorylation means we're going to take an ADP and we're going to take a phosphate group off something that's hanging around, some substrate, an inorganic phosphate, and we're going to use that to make ATP. Okay, so this is substrate level phosphorylation because we're just ripping it off something else that's in the cytoplasm. From there, these pyruvate molecules are going to move into what? What is this thing? Yeah, so this is, from there, we're going to move into the mitochondria. And technically, it's a mitochondrion because it's only one, but we always just generally say mitochondria. And there's several things that are going to happen here. So the next step, and some people kind of put these two things together as number 2, is pyruvate oxidation. What does that mean? Oxidation is losing electrons. So this pyruvate is going to lose electrons. We're rearranging the carbons. And we're attaching a coenzyme. We're making acetyl-CoA, which is 3 carbon. Again, we're not worrying about the details of it yet. From there, we're moving into the citric acid cycle. So this molecule, this acetyl-CoA, goes into the citric acid cycle. It's going to produce a little bit of ATP. Again, this is just substrate level. phosphorylation. Phosphorylation, again, just means we're adding a phosphate group to an ADP, which is adenosine diphosphate. We're making adenosine triphosphate. We're adding a phosphate to do that. And we're also going to fill some electron carriers. So we're going to have NAD plus is going to become NADH, and FAD is going to become FADH2. So we're going to take these electrons and we're going to dump them into this last stage again too. So that's the second or second and third, depending on which textbook you use. And then the last phase is where we get the big payday in terms of ATP. It's oxidative phosphorylation. So what does oxidative tell us is happening to electrons? We're losing a lot of electrons, and phosphorylation tells us we are making ADP into ATP. So we're losing electrons. In the process of losing electrons, we're taking ADP to make ATP. We're adding that phosphorylation group. This is a two-step thing. The first step is the electron transport chain. And the second step is chemiosmosis, which we're not going to talk about all the details of that yet, but this produces a lot of ATP. You'll notice this is a little bigger than the other ones. This oxidative phosphorylation process produces lots of ATP. This whole process overall total produces about 32 molecules of ATP. About, because it does vary some, but it's the average is 32 molecules of ATP that are produced by this process. And that is where we'll stop today. And we will get into the details because we haven't done the details yet. On Wednesday. Because you're going to go sign up for the Beyond Conference and you're going to come to the Adopt a School meeting at 11.